So in this video, we're going to talk about the stomach. So first off, it can be a storage container for food that's been eaten. This is because the stomach has kind of larger size and can be stretchier than the small intestine. So if the small intestine is still working on digesting food, there's only a certain amount that can go and stretch into the small intestine. So if you're waiting for food to kind of continue down the line, to continue down the small intestine, food can sit in the stomach for a little bit and be stored because it can stretch out a lot more. There's also two types of digestion that occurs in the stomach. One is mechanical, meaning that it just kind of hammers it down. So again, you have a food bolus that's coming in originally. This is still too large for enzymes to break down quickly. So what do? We're going to break it down into more of like a paste. And that's done by mechanical digestion because you're just kind of, it's like kneading bread or bread dough. So you just kind of break it down and break it down and break it down. So the stomach's going to knead this food bolus until it's a paste. And it's a paste called chyme. And so then we have chyme that's going to then go into the small intestine, which can be more kind of easier to handle. We also have specific chemical digestion. This isn't happening for all food, but we can break it down with acids. So again, hydrochloric acid is going to be breaking down the bolus even further. So you're breaking it down with a paste by just kind of mushing it and mashing it, and you're also breaking it down with chemicals, so acids and enzymes that we'll talk about. So protein digestion, especially enzymes for proteins, is going to start here. Stomach also produces intrinsic factor. So we're going to talk about intrinsic factor a couple of times, but intrinsic factor is needed to absorb vitamin B12. So we're going to learn about this more when we get to the large intestine, but without intrinsic factor produced by the stomach, we can't absorb vitamin B12. And vitamin B12 is going to be made by the bacteria in the large intestine. And remember, B12 is used for cell division, so mitosis. So cells that are dividing constantly need vitamin B12. So things like red blood cells, white blood cells, intestinal cells, hair cells, skin cells, all these things that are dependent on mitosis and dividing constantly need vitamin B12, and to absorb it, you need intrinsic factor. So I'm looking at the stomach, you always have some volume in the stomach, about 50 mils of volume. So this is milliliters. And then it can expand up to 4 liters. So this is when I say that it's really, really stretchy. It can expand a whole lot. Not that you want it to, but it can. It's stretchy. And its muscularis externa, the outer muscular layer, actually has 3 layers of smooth muscle. So we saw in the esophagus and talking about peristalsis, we saw longitudinal and circular, which is going to be what's in small intestine as well. So this is going to be throughout is circular, so going kind of around the outside, and then longitudinal going lengthwise. But then with the stomach, we have an oblique one, so it's at kind of a weird angle. So this is just going to help with moving and churning the stomach. So this is actually able to mix around. Once you get a food bolus, you're going to see how the stomach kind of mushes things around coming up. So you get a food bolus come down. Well, all the muscles in the stomach can kind of slosh it around and break it down physically because it's squishing here, contracting here, moves it back, contracts here, moves it forward. So you're sloshing and again breaking it down, similar to what we learned about with segmentation. It's also mixing. So you're mixing those enzymes, you're mixing the hydrochloric acid, and you mix what we say again, chyme. Once you're breaking down that food bolus, we call it chyme, and that's what's going to enter the small intestine. So it's kind of like a paste material. In the mucosa, or the inner lining of the stomach, has gastric pits, and they lead to glands. So this is just showing you have the mucosa, and then there's a kind of valley, and then the glands are going to be in the valley. So you can see here, this is the gastric pit, and then you have glands down here. So you have glands that are going to be deep in kind of the side of the pit. These glands are producing what we call gastric juice. So it's going to be the liquid that's added to the stomach to help break down food as well. And we have 4 types of cells or glands. There's parietal cells, not to be confused with things in your skull, your parietal bones, lobes. You have chief cells, you have G cells, and you have D cells. So first, we're going to talk about parietal cells. Parietal cells make hydrochloric acid. So again, hydrochloric acid, pH of about 1 to 3, 1.5 to 3.5. So very low pH, very acidic, very damaging. It denatures proteins. Remember when we talked about proteins in AMP1, that proteins have specific shapes. So you have your primary, which is your amino acid chain. You have secondary, which is your alpha helix and your beta pleated sheets. And then you get to more folding, so more complex folding and tertiary and quaternary. So proteins have to have a specific folding pattern to them to function appropriately. Hitting this hydrochloric acid makes them unfold. Now, this is a good thing when you want to digest the proteins, because it's easier for enzymes to break them apart when it's a straight line and they can just kind of break them versus getting in there when it's all intertwined. So unfolding or denaturing of the proteins actually helps with breaking them down. Also, plants, animals just have their plasma membranes that we talked about. Plants actually have an additional kind of protective wall around their plasma membrane called cell walls that can make them harder to digest. Bacteria sometimes have them too. So this hydrochloric acid can break down this cell wall that's extra protective to get to the actual cell, that with just the plasma membrane, that's easier for other things to penetrate. So cell wall is just thicker. The hydrochloric acid, again, is going to help kill bacteria. So immune function is going to be, when we see acids, a lot of them are going to be helping with the immune system as well. And then we have activation of pepsinogen. So we're going to look at chief cells next. And chief cells make pepsinogen. But we need pepsinogen. So when you have pepsinogen, that is a protein, a proenzyme that's going to break down proteins. Well, we release it in the inactive form because there's a lot of proteins in your body. You don't want an enzyme to start breaking down until it gets into the stomach so that it can break down the proteins that you're eating, not the proteins of your body that are supposed to be there. So pepsinogen is made by chief cells, and we'll talk about those next. And it needs to be activated, and it's actually activated by the hydrochloric acid to then be activated to pepsin. So without hydrochloric acid, you just stay pepsinogen. So you stay inactive. You don't do anything. It's not pepsid, it's pepsin. It's not the antacid. So pepsin is going to be the active form, and that's going to actually start breaking down proteins. So you have to have the hydrochloric acid so that you can activate, so that pepsin can work. Mention intrinsic factor already. I'm going to mention again. That's where it's made in the stomach, is by parietal cells. So parietal cells are making intrinsic factor so that you can absorb vitamin B12, anything that you're taking, or also that's being made by your large intestine, anything that you need absorption of B12, then it's going to need intrinsic factor. You need both so B12 can be absorbed and used. So again, just mentioned it, but I'm going to mention it again. Chief cells are making pepsinogen. They're not making pepsin. They don't make pepsin. The only time you get pepsin is when pepsinogen hits hydrochloric acid. So pepsinogen hits hydrochloric acid, pepsin. It makes pepsinogen, the inactive enzyme. It's activated to pepsin by hydrochloric acid. So if the chief cells didn't have kind of no exposure, so chief cells make pepsinogen, there's no exposure to hydrochloric acid, pepsin never happens. Pepsin, pepsinogen, once it's activated to pepsin, is a protein digesting enzyme. So that's why you don't want it activated again, because you want it to actively break down the proteins that are in your stomach. You don't want it to break down the proteins that are in your body naturally. We also have chief cells that make lipases. Lipases are going to be fat breakdown enzymes. So ases is an enzyme. Lipases is fat breakdown. This is going to be a small amount of fat, so about 15%. And these again are milk fats. So it can start with a salivary lipase or lingual lipase and start breaking down fats. These are easier to digest. They're not kind of the normal fats that we think of. That's again going to be in a different process. So we have G cells in the stomach that are going to produce a lot of hormones, and these are going to be paracrines. So they're not ones that are going necessarily throughout your entire bloodstream. It's going to be more local hormones. Major one is going to be gastrin. Gastrin's a hormone that's actually going to stimulate the parietal cells and the chief cells. So this is going to help, this is going to be around when you've eaten something. So gastrin's release, you start producing hydrochloric acid. So you can start helping break down and activate pepsinogen that's being made by the chief cells. So this is going to help. We don't need secretion of hydrochloric acid and pepsinogen all the time. We just need it when we're about to digest something. It can also stimulate smooth muscle contraction in the stomach. Well, part of the stomach's process isn't just breaking down with enzymes, it's the mechanical breakdown, and that's done by smooth muscle. So you're starting to contract your stomach. So gastrin is a hormone that's getting and communicating to your stomach, hey, food's coming. You need to start getting ready for digestion. D cells in the stomach produce a hormone called somatostatin. That's going to inhibit G cells. So if you're inhibiting G cells, you're inhibiting digestion. So you're stopping digestion. So this is when stomach's empty, you haven't eaten in a while, you don't need to be producing enzymes like pepsinogen. You don't need to be producing hydrochloric acid because you're not eating. There's nothing to digest. Once you inhibit D cells, which are inhibiting G cells, that's when G cells are allowed to produce gastrin and among other hormones, and it allows digestion to occur. So D cells are saying don't digest. Leave it don't. And G cells are saying go. Do digest. So G for go, D for don't. Think of it that way. So go is go digest. D cells don't digest. Looking at the peristaltic waves in the stomach. So this is going to be forward movement. where we need to kind of measure the amount of fluid that's going in the small intestine. Remember, we can stretch the stomach out a lot. We can't stretch out the duodenum or the beginning of the small intestine a lot. So we need to control the amount of material that's going into the small intestine at a time. It and kind of slowly digest it. So first things first is the peristaltic wave. So it moves from the fundus, which is this upper part. So this is the fundus of the stomach. This is the pylorus or the pyloric sphincter. So we have the pylorus and the fundus. We have movement from, and this is where the esophagus is entering. So we have movement from the fundus towards the pylorus. Contraction of smooth muscle. Then we have a valve. So we have a little kind of indentation and a little valve of smooth muscle called the pyloric valve. So pylorus, and we have a little opening, and this is going into the duodenum. So what happens is the smooth muscle wave comes down and goes to the pylorus, and we get the valve open a little tiny bit, so a little bit of chyme can enter the duodenum or the first part of the small intestine. Doesn't open a lot, so a little bit of chyme squirts out, goes in the small intestine, goes in the duodenum to continue to be digested. Then that valve just opens a slight, lets a little bit of material out, and then it closes back again. So then all of this wave, so we have the wave coming down, a little bit goes through, and then this closes. So once this closes, all of the material that's flying down towards bounces back because it's hitting a wall. Can't go anywhere. So the peristaltic wave closes the valve, and then we get it bounce backward towards the body of the stomach and back towards the fundus. And that's called retropulsion. And then that continues. So then we have a peristaltic wave, start again, opens the valve, a little bit squirts out, and then swishes back. So that's why we slowly empty the stomach, because again, it makes it more manageable for the small intestine, for the duodenum to handle it. Instead of having all this material, you can't put all that material in the small intestine, and it's just going to slide through. So we need small amounts, small manageable amounts. So this is how that happens.